Inside the inner membrane of mitochondria, electrons flow down 4 protein complexes, creating a proton gradient that drives ATP synthesis. These 4 complexes form the electron transport chain. Coenzyme Q is a molecule that transfers electrons and protons between complexes. Coenzyme Q has 3 states: the oxidized ubiquinone, the partially reduced radical semiquinone, which carries 1 electron and 1 proton, and the fully reduced ubiquinol, which carries 2 electrons and 2 protons. These protons contribute to the decrease of the proton concentration in the matrix. Complexes 1 and 2 harvest electrons and protons from NADH and FADH2 respectively, reducing coenzyme Q. Ubiquinol released by complex 1 and 2 diffuses in the membrane and binds to complex 3. Ubiquinol is reoxidized by transferring its electrons and protons to complex 3. The electrons accepted by complex 3 are then transferred to cytochrome C, bound at the surface of the membrane. One of the challenges during electron transport is the transfer of electrons from ubiquinol to cytochrome C. While ubiquinol carries 2 electrons and 2 protons, cytochrome C can only accept 1 electron. Complex 3 solves this problem by transferring the 2 protons into the intermembrane space and transferring 1 electron directly to cytochrome C, while the other one is temporarily housed by complex 3. Where does complex 3 store the second electron? Complex 3 controls the divergent path of electrons in a process called the Q cycle. The Q cycle is a 2-step process involving the oxidation of 2 ubiquinol molecules and the reduction of 1 ubiquinone molecule. The result is a release of 4 protons into the intermembrane space and the reduction of 2 molecules of cytochrome C. Complex 3 contains 2 binding sites for coenzyme Q, Q0 and Qi. First, a ubiquinol molecule binds to the Q0 site of complex 3, while a ubiquinone binds to the Qi site. Ubiquinol is oxidized, and the 2 protons are released into the intermembrane space. Of the 2 electrons, only 1 is immediately transported to cytochrome C via a series of electron transporters. First, a risky iron-sulfur protein, then cytochrome C1, and finally cytochrome C, bound to the intermembrane site of complex 3. Once reduced, the cytochrome C dissociates from complex 3. The second electron from the Q0-bound ubiquinol is transferred to a series of electron donors, the Bl heme, the Bh heme, and finally ubiquinone on the Qi site to create a radical semiquinone. In doing so, the semiquinone takes a proton from the matrix. In the second step of the cycle, the ubiquinone bound at the Q0 site is replaced by a new ubiquinol, while Qi is still occupied by its semiquinone. As before, the Q0-bound ubiquinol releases 2 protons into the intermembrane space. 1 electron reduces another cytochrome C, while the other electron and a proton extracted from the matrix complete the reduction of the semiquinone to ubiquinol. Complex 3 returns to its initial state when the Q0 and Qi binding sites release their respective coenzyme Qs. Complex 3 is then able to begin a new cycle. Complex 3 not only solves the problem of electron transfer between ubiquinol and cytochrome C, but it also contributes to the building of the proton gradient across the inner membrane of mitochondria, fueling ATP synthesis. Cytochrome C shuttles the electrons from complex 3 to complex 4, where these electrons will reduce oxygen, the final electron acceptor.